Um, I'm now recording. Okay, maybe we can start.
Okay, hocam.
Yeah, Irem is gonna present. She's fourth year bachelor student. She came to the lab meetings, I think, like 4 or 5 weeks ago, I guess.
Hocam, more than that, as far as I remember.
Really? Okay. Uh-huh. Yes, and then let's hear her presentation. So she took a high responsibility. The paper she's gonna present is having a lot of molecular biology. Although her background is biology, she put a lot of effort, I think, to understand what these figures are telling to us.
Yes, hocam.
Mhm. Okay.
Then I'm starting.
Yes, please.
Today,
Yeah. The stage is yours.
thanks, hocam. Today I'm going to talk about the article named as "The Transcription Factor BHLH121 Interacts with ILR3 and its closest homologs to Regulate Iron Homeostasis in Arabidopsis." So as we all know, iron is a very important micronutrient, both for plant growth and development, and it also affects plant productivity and plant product quality. So in this article, their main aim was to demonstrate the effect of BHLH121 on iron responses. So in first figure, their aim was to demonstrate possible in vivo interactions between BHLH121 and ILR3 along with its closest homologs. To show this, they first conducted yeast-2 hybrid assay. I just wanted to give you a little bit background information about this assay, although we talked about this a lot. So in yeast-2 hybrid assay, we have a transcription factor. It's composed of two domains, activation domain and binding domain. When this transcription factor binds to the upstream activating sequence, it will just activate the reporter gene, which is in this experiment history gene. So what should we do is we should split this transcription factor into two so that activation domain and binding domain will be exposed. And we will just fuse our proteins of interest to binding domain and activation domain. And these proteins will be named as bait and prey. And both of them will be present in different plasmids, as you can see here. So at the end, if the yeast takes up both of these plasmids and also our proteins of interest are interacting, this transcription factor will be intact and it's able to just activate the reporter gene, which is histidine-3 gene here. After that, to visualize the interacting proteins and yeast, we should just use a media which lacks histidine along with tryptophan and leucine. So if we come to the figure 1, here binding domain alone used as the control group. As you can see, there is no signal here. So here, our protein, BHLH121, is bound to the binding domain and other proteins are bound to activation domain. So here it's non-selective medium. It lacks tryptophan and leucine. However, here our selective medium and it lacks histidine too. So this chemical is called as 3-amino-1,2,4-triazole and it's competitive inhibitor of the history enzyme. And this is used whenever the history gene is used as the reporter gene. So this is used to just inhibit the background growth. So there we see the background growth here, for example. We don't want any background growth because we may easily presume that, for example, BHLH121 interacts with BHLH11 or with itself. But this is false positive result. So if we add this chemical in increasing amounts, we will just inhibit the background growth here. So when we look at the results, we see that BHLH121 is able to interact with BHLH34, 104, ILR3, and 115. However, it will not interact with PYE, BHLH11, nor with itself. So is there any questions here?
Yes. Why do they choose these transcription factors but not the others?
Because these are the closest homologs of ILR3, hocam. They are just wondering whether this transcription factor is affecting this one. This is the crucial one. And these are the homologs of it.
Okay. How do they start working on BHLH 121. Any idea?
Hocam, I honestly don't know. I didn't check for it.
So I think ILR3 was already known. It's a transcription factor. Everybody knew it's above fit, and it's a very important one, but nobody knew with which transcription factors it's interacting with. So what they did was they already have a ILR3 tagged mutant, and then they did co-immunoprecipitation, co-IP. So they crushed the plants and used a column and then capture the tagged ILR3. And then they have ILR3, but it's not just pure ILR3. If anything binds to that, all these proteins will also come, okay? And then they used ICP, no, not ICP, MS methods to identify which proteins are in the mixture with ILR3. Sorry, with ILR3. And then BHLH 121 came.
Thanks, hocam.
İrem, I have a feeling you're a bit excited.
Yes,
Were
hocam.
you able to listen to what I said, or did you just were you just talking, thinking about what are you gonna tell next?
Yes, hocam. I actually feel nervous because I'm afraid that I sometimes forget something that I say, and it's a very important thing. So I just sometimes talk too fast. If that happens, just like before, just warn me, please. So I will try to slow down.
Okay.
I actually have post-its all around me telling us talk slow, but it doesn't work so well. So okay.
So now about this situation, İrem. Actually, in the first section, in the first paragraph of the results, they are explaining how they found BHLH121. So if you ask to me first, before showing this yeast experiment results, I would just show the supplemental table 1 in order to tell what type of experiment they did and how they found 121.
Okay,
so after showing that, showing this yeast experiment result would make the entire story connected.
Exactly. And when I was in Marilou's group, Marilou was asking almost in every presentation, how did the story begin? Yeah, so it's like a storytelling, giving a presentation, and the background information is this. How you tell some information, and then at the end of the background you tell how the story started.
Okay, hocam. Thanks for
Good.
feedback. In figure B, they just wanted to further confirm what they have found in yeast to hybrid assay, and they have conducted the bimolecular fluorescence complementation assay. The difference between yeast to hybrid assay is this method is done in vivo, exact location. So what we do is the proteins of interest will be fused to non-fluorescent part of the fluorescent protein, which are in N-terminus and the C-terminus parts. And if those two proteins are interacting with each other, these two non-fluorescent part of the protein will be brought to close proximity, and we will see a signal so that we can say that these two proteins are interacting with each other. And if they don't, we won't see any signal. So here, this part is used as the control group, and here we can see the positive results, which are correlated with what we have saw in the yeast to hybrid assay. So here in control group, if you realize that BHLH121 is also fused to N terminus and the C terminus in both of them. So this is just to check whether this protein forms any homodimers with itself or it's able to self-activate itself. And as we can see here, it's not. And it's able to form homodimers with BHLH34, 104, ILR3, and 115 again, but it's not able to form any heterodimers with PYE and BHLH11.
Sorry.
Yes.
Before continue, can you explain what this image is representing? I mean, those individual oval shapes, what are they? Where did they express these genes?
Hocam,
what is this big circle there?
Yeah. What's it?
The big circle, like that looks like an animal cell.
Hocam, I actually tried to find what they means, but I couldn't find, and I just passed it. So I don't know what they individually represent and why they merged. I also don't know this, for example. I just know the results.
Hocam, aren't they looking all the components of the system separately at first and then checking whether they're all expressed together at the end bar, the merged bar?
Yes. The question is, what is this circle here?
Do you think it is a cell or something different?
It might be a cell. It looks like a cell, but I'm not sure.
What cells do you think is it?
Maybe Arabidopsis protoplasts.
Okay, protoplast, very good. How did you understand that it is protoplast?
Hocam, because if it wasn't a protoplast, I would say I would saw a more rectangular or square type of cell maybe. But if the cell wall is disrupted, it will just shape like this with the cell membrane.
Okay, good. Yes, you are true. This is a protoplast, and they are actually expressing the two vectors in the protoplast simultaneously. And if there is interaction, then they give this green color. And actually now on the left side, you have the YFP image. It is representing the interaction, and they take this image. If they, I don't know what type of microscope they used. I think it is a confocal microscope. In this confocal microscope, you can just use the green channel for the YFP signal. Then you can take the bright field image under the normal light, like compound microscope, and then you can take the image in the red wavelength for the chlorophyll autofluorescence. Then you can merge all of these together in order to prove that the interacting YFP signal is not coming from the chlorophyll or chloroplasts, but this interaction is somewhere very specific in the cell. Where do you think it can happen? That green part, where do you think, which organelle is it?
Which organelle? I'm not sure, hocam.
No, definitely it is not chloroplast.
Yeah.
Because signal is not coming, it is not merging on the red one. Do you think that it is dispersed into the cell?
Maybe ribosome?
No.
Should it be dispersed? I don't know.
It is not dispersed. If it was dispersed, you wouldn't be able to see individual dots. It would be everywhere. So definitely it is not in the cytosol. It is definitely in a compartment. But what is this compartment?
It looks like there is only one of this compartment in the cell.
Yes, absolutely.
I would say nucleus, but I'm not sure.
I was just about to say nucleus, but
Okay, now perfect answer, but why?
I also don't know for 100% fact on this. I don't agree on it, it might have a tag of a specific kind that specifically like goes to the nucleus because it has information there. But I don't know.
Okay, going in the good way. Now we are in the track, but now when you look at these proteins, what are they?
Because they are factors, so they will bind to the DNA.
Yes, they are transcription factors.
Yeah.
They are only found in the
nucleus.
What is that? Nucleus. Therefore, these green dots are only coming from the nucleus.
Yes, transcription factors bind to DNA, and DNA is in the nucleus. That's why.
Thank you. Bye-bye.
Should I pass this?
In fact, I have a question. How do we transfer the vectors to the cell? Do we use agrobacterium? Emre Hocam?
In this experiment specifically, no. You cannot. Here they use PEG.
I see. So we just make the cell competent. Right?
Now here actually you are increasing you are changing the osmotic pressure so that there will be little cracks on the membrane, and then your DNA, these vectors will enter. This is one way. I think they use this way. Or another way is using the electroporation. You can give small, short period of high electricity, and then it can also make crack.
Okay. I have another question. So the difference between this one, I mean this experiment BIFC, and the yeast experiment, I think. I mean, there might be many differences, but one difference should be in the yeast experiment, if there is a complementation, then it should mean two proteins are directly interacting with each other. There are no other proteins needed. But if we see there's interaction here, it might be the case that the two proteins are not directly interacting. Maybe there are other proteins in between. Do you think I am right?
It is possible here.
Yeah, that's because now there are a lot of different proteins because this is plant cell. In a plant cell, there are a lot of different proteins interacting with these transcription factors. So when we see interaction, it shouldn't necessarily mean the two proteins directly interact. Maybe there is a bridging protein. But in yeast, since yeast is a completely different organism, you don't expect, I think, interaction between yeast proteins and the plant.
True.
Okay. Did anyone get the point? Anyone who could follow?
Yeah, I got the point, but I couldn't conclude that. Is it because it looks smaller than the others? Because of this reason, it has the other proteins?
No. The reason is if you do the experiment in plant, then your proteins are not isolated from the environment. There are a lot of different proteins which can interact with your protein. But if you do the experiment in yeast, since there are no plant proteins other than the ones you express, if they interact, it must be direct interaction.
Yeah. So do you think that if they did this experiment first and then the yeast to hybrid assay as a confirmation, would that make more sense?
No, because I am just talking about the nature of these experiments. There might be other reasons why they choose this order but not the other.
Now, if you understand, basically, in all papers we have seen, always they show the yeast experiment first, and then the other types come. The reason for this is for the yeast, it is expected, it is similar to a an in vitro experiment.
Mhm.
And for instance, in this YFP split luciferase or something different, it is found in the plant cell or plant tissue. Therefore, it is counted as in vivo. But for instance, this one is not real in vivo. Split luciferase is almost in vivo, but the real in vivo is the co-immunoprecipitation experiment.
So now, please listen to me carefully. You can have two approaches in biology. One, you can isolate the thing you're searching for. The second, you don't isolate it. You look how it is working in where it is. So if you take the first approach, you isolate it from all the environment, then it gives you very direct information about the nature of this thing you're searching for. But you have the disadvantage that it may not behave like that in the real system. But if you work in the real system in the plant, then you have the advantage. You know how it works in the real system, but then you have the disadvantage. Whatever you measure, you find information about. It might not be direct consequence of your the consequence of the nature of your protein. Maybe there are some other interacting partners. So your result is like a harmony of the results of all these interactions and so on.
Should I continue, hocam?
Yes.
In figure 2, their aim was to examine BHLH121 loss of functions, mutants, to see whether their iron deficiency tolerance has been affected or not. So first here, they just plated them to agar, and they see that, for example, when iron is not present, mutants have severe growth defects than compared to wild type. And in control conditions, wild type again grows better than all of these mutants, and the mutants have observable chlorosis symptoms. When we apply mild excess iron conditions, they are indistinguishable from each other. However, when we apply excess iron conditions, we see that mutants grow better than the wild type. So just a note, all of these three mutants affect the integrity of the BHLH DNA binding domain, so they have similar effects.
Okay, sorry, before we continue, what is the meaning of DNA binding, integrity of the DNA binding domain? Why do you think this is critical?
Hocam, because these then transcription factor cannot bind properly and it won't be very effective.
So they don't have the real function, right?
Yeah.
Okay, and now can you tell us how they generated these mutants?
Hocam, I actually write that down, but I have to look at my notes for this.
Please look.
They are generated by CRISPR-Cas9 system, and one of them was actually truncated protein, but I don't remember the other two.
I think one of them is a deletion. I actually read, but I forgot. One of them is deletion, other one is another deletion, I think.
And the last one was just a truncated protein, early cut protein.
Now, so basically in Arabidopsis, there is, you can pay money to buy mutant seeds. And these mutants could be tDNA insertion lines. Why do you think that the authors, in this case for the BHLH121, didn't buy the tDNA insertion mutant lines they just wanted to generate their own mutants through CRISPR-Cas?
Hocam, because when the tDNA method is used, it's just, how can I say, just injects its genome in different parts of the other organism, in, for example, Arabidopsis, and it may create other defects in the genome of Arabidopsis. So maybe I wanted to just look at the mutants in one point, for example, but the result might be just stemming from other alterations too. So they just created their own mutants.
Now, I like how you are thinking, but I need to clarify something about the tDNA insertion lines of Arabidopsis. Basically, yes, you are right. After adding this tDNA into the plant genome, that it can be integrated in several places, but then they do a back cross in order to eliminate the unwanted mutations. So basically, the seeds that you buy are pure homozygous tDNA insertion lines that only has almost, you will never be 100% sure, but almost clean. The rest of the DNA is almost clean of the mutation. But still, they didn't want to use those tDNA mutants, and they generated their own CRISPR system. By the way, in CRISPR, when you use it, there is a big chance of off-target mutations to happen in other parts of the genome.
I didn't know that.
So even if there is this problem, they still insisted on using this technology. Can you guess the reason why they did this now? Do you think that they wanted to use a fancy technology?
Hocam, I was about to say this, for maybe just they just wanted to just say I used a very good method for this to create the mutants, and I didn't buy them. I didn't just use easy way. Maybe I don't know actually.
Seçkin hocam, what do you think?
Yeah, I know the answer. So of course, there wasn't any tDNA insertion mutants for this gene. That's why they have to produce themselves.
Yeah.
And that's also a reason why this very important, very critical gene has not been found before. So if there were tDNA insertion lines, they would come up from the screening approaches, I think, very easily.
Yes. Thank you very much.
I'm passing this.
Yes.
In part B.
Just a second. If something is not clear for anyone, you should ask for clarification. I really don't know if you can follow up or not. It's your responsibility to understand, and the reason we do this, spending all our time, is not to do it and finish it. It's for your education, and if you are not understanding, then it's a waste of time. You should ask.
Hocam, can I ask a question
Yes,
about
please.
this figure? Okay, for the 500 micromolar iron part, yes, that part, the wild type exhibit the defective growth. Is it because since they can carry a normal iron uptake, are we seeing an iron intoxication here? But since our mutants cannot carry out a normal iron uptake, they can be they're showing a better phenotype. Is that why?
So your question is why mutants show better growth in high
Yes.
iron? Yeah, any ideas?
Was my explanation right?
Your explanation, can you tell it again more clearly?
Okay.
Maybe?
Okay, the wild type, our wild types are carrying out a normal iron uptake. So in this excessive iron conditions, they are intoxicated by irons because there's a lot. But since our mutants cannot carry out a normal iron uptake, they can tolerate this better than the wild type. So that's why we're seeing this kind of growth. That was my explanation.
Yeah, I think mutant is unable to take up iron as much as wild type. And if you put too much iron, the one who is unable to get iron will be advantageous, I think.
So,
Should I? Okay.
So that means the mutants should have been grown better, isn't it? That means that because they can carry out a better iron uptake.
they can't carry out a better iron uptake. So let's see. So the mutants cannot take up iron as efficiently as the wild type.
Yes.
And if you put too much iron, then the ones which is unable to take up iron will be advantages.
Okay, that's what you're saying. Okay, I understand.
Yes, because this is too much. This is bad for the plants. If you have too much of a bad thing, if you're unable to take it, it's advantages.
Okay, so my guess was right, I guess.
Yes, but maybe you could explain it more clearly. I mean, it was not that clear to me. I don't know the others. Maybe we can give an analogy. So let's assume there is a guy who is unable to eat as much as a normal guy, okay? So in normal conditions, doesn't matter. If there is a lot of food, doesn't matter. The guy who doesn't eat so much will still develop like the normal guy. But if the food is too less, then the guy who cannot eat too much will have a problem. But if the food is excess, then the guy who cannot eat that much will have an advantage. I don't know if it explained better.
I think a different analogy might be how like different people are accustomed to different temperature. So for example, people who are very disadvantaged because of really, really freezing cold temperature, like the people who live in Siberia. But like when temperatures are really, really freezing, those disadvantaged people are now the advantaged, but it's a rare occurrence. But in that case, then they would be the advantaged people.
Yes, thank you very much.
Yeah,
Analogy.
yeah.
Hocam, I'm passing it.
Just a second. Babar, we were talking about how much iron should we put to generate iron toxicity?
Yes, and they are using 500
Yes.
micromolar.
Okay. Yeah, you can pass.
Hocam, in part B, they just compared the root length. Actually, this is just what we saw here. They just confirmed what we saw here. So I'm not going to go over this. In part C, we see the micromolars of iron 2, which is the indicator of ferric calase reductase activity. And this activity, ferric calase reductase activity, is induced under iron deficiency. So when iron is not present, wild types FCR activity is higher than the mutants. However, in control conditions, their activity is not very different in wild type and mutants. When we compare the fresh weight, we see that in iron deficient conditions and control conditions, fresh weight of wild type is higher than the mutants. However, it's way more higher in the control conditions. When we apply mild excess conditions, we see that fresh weight of wild type and one of the mutants are not that much significantly different. However, wild type is lower than the other two. And in excess iron conditions, wild type's fresh weight is lower than the mutants. When we look at the chlorophyll content, again, it's higher in wild type for iron deficient control and mild excess conditions. However, it's in iron excess conditions, wild type and mutants does not differ that much in chlorophyll content. In graph F, we see the iron content directly. So when iron is not present, iron content is not significantly different in mutants and wild type. However, in control conditions, wild type's iron content is higher than the mutants. So is there any questions?
Yeah, the problem here is you described us the data, but you didn't give us any meaning out of it.
Yeah, I didn't.
Okay, I think you can pass, but just a note.
In figure 3, they aimed to grow mutants on soil to observe their phenotypes and visualize the iron content and iron accumulation in their seeds. So in part A, they just compared the control conditions and iron present conditions. Iron is applied exogenously. In control conditions, wild type grows better than all of the mutants, and they have severe growth defects besides the chlorosis symptoms, as expected for this mutant. When the iron is applied exogenously, all of these mutants' phenotype are rescued. Then they just compared the chlorophyll content of these.
What's the reason they do this experiment?
To just check whether BHLH121 affects iron responses, iron uptake responses.
But didn't they do the same in the previous picture?
They did this for soil now, so they just try to mimic the real conditions, maybe.
Okay, this is the important point. So you should start you should not tell us this happened, this happened. You should tell us now they wanted to see if what they see in the agar medium holds true for more real conditions. So this is the meaning.
Okay.
Okay.
In part B, they just compared the chlorophyll content again. In control conditions, wild type's chlorophyll content is higher than all of the mutants, as we can see here. And when iron is applied exogenously, the chlorophyll content is similar in wild type and the mutants.
Now, one thing in this B graph, normally the total chlorophyll content is shown in the unit of microgram per gram leaf fresh weight. Here they just did the calculation according to the area. I think this is interesting. I never seen this before.
Yeah. It's very interesting.
So maybe the fresh weights were same or something different. When they did the calculation with the fresh weight, maybe they didn't see a good difference. That's why they are using the centimeter square.
I guess this should be a simple mistake.
I don't know. I was just looking to the methods. They said that in some experiments, they showed it in fresh weight. In other experiments, they had a disc, leaf discs, and then they calculated the area of that disc.
Wow, okay.
Interesting.
Oh, okay. Yeah, I think they may have a machine which can make a disc from the leaves. I don't know. Okay.
In part C, they just grew the seeds in soil for 6 weeks, and again they just observed the similar fashion what they have found in part A. So as we taught here, mutants' phenotypes are just rescued again when iron is applied exogenously.
Yeah, what's the reason to do this experiment?
Maybe they just want to check the long-term effects of this mutant and applying iron.
Yes. So this is the important information. Nothing
I
else.
should give that first. Okay.
Yes. Okay.
In
Also,
part B,
in there they also actually I asked it early. Can you tell the other slide, please? Yeah. No, you can continue. I asked it a little bit early. I will ask it a little
Next
bit.
slide.
You are talking about this part?
Yeah, you can talk about this part after, then I will ask a question.
Okay, in part D, they just compared the iron content in seeds of wild type and the mutants, and they see that wild type's iron content is higher than all of the mutants. And in part E, they just wanted to check the iron accumulation in the seedlings, which are 4-day-old. And as we can see, wild type is able to accumulate iron. This experiment is done with Persdap staining. However, mutants are not. Mutants seem like they just don't able to accumulate any iron. So maybe their iron uptake machinery might be impaired due to loss of this transcription factor, this gene. Yeah.
Why they did this experiment?
Just to check if mutants affect the iron accumulation in these plants.
Yeah, Uh-huh.
in seeds.
In seeds, sorry.
Yeah, it's in the seeds, and these are the growing seeds, I think. Also, I didn't get that why they need to look the seeds, because I think it's expected to see a little bit lower iron content in the seeds in the mutants, because they cannot get enough iron from the soil or agar, the environment.
What?
I didn't get why they need to look at the seeds for iron content, because it is very expected if the plant cannot get enough iron from the environment. Of course, there will be a less iron in the seeds.
Mm-hmm. Would there be another possibility? Would anyone from you expect a similar level of irons? Or do you agree with Onur?
I don't think there's a strong correlation with the iron uptake ability and the seed iron accumulation levels, because in some cases, when the plant is suffering from iron deficiency, it can accumulate all of its iron on its seeds for survival.
Mm-hmm.
So I kind of understand why they checked the seed iron contents.
Okay.
Yeah, so Onur said it's very usual and thus not needed to look at the iron concentrations in the seeds. But I think a guy who is long in the field would know that even iron deficient plants accumulate same amount of iron in the seeds normally. And the reason is not known. So usually doesn't matter how deficient the plant is, maybe the number of seeds decreases as you see in the picture, in the above picture. Can you go one slide less? One slide back. Yes, here if you, no, here.
Ah, okay.
Not here.
This one?
Yes. So here you see there are a lot of seeds. Here you see there is very little seed. There are very little seeds. Usually, if it is like IRT1 mutant, for example, the seed iron level is almost the same as the wild type. That's why they want to look in the seed. And what they found is really seed iron level is very low.
Okay.
Babar, do you think it's interesting result for us?
Yes, it is.
Why?
Because if the plant is not able to uptake from the soil and it is there is less iron in at vegetative stage, how come it accumulates a lot in seeds? Is it coming via cells or some other transporters that are not interacting with this gene?
Yes, next slide, Irem, please. Okay. Yes, so Emre Hocam, what we hypothesize is if a seed has less iron, it germinates later.
It does.
But we don't know.
Oh, really?
Yeah, it's not established. So Babar, what we can do is we can also order these seeds and look if they germinate also later.
Okay.
We did one small experiment. We actually we grew different soybean genotypes in iron deficient, I should say, high TH condition, and then we collected their seeds. And then in the next generation, we grew those seeds in iron plus, iron minus conditions. And actually, plants become more sensitive to iron deficiency in the next generation if the parent is grown in iron deficient condition. So this and this comes because of their less iron accumulation in their seeds. The parent accumulates less iron in the seed, therefore those seeds grow much slower and more sensitive to iron deficiency.
Yeah, so these field crops, they give different responses. Some of them under iron deficiency, they still have the same level of iron in the seeds, but some has less. In your case, it was less, but it's like a debate, you know, why some plants have less. Usually the seed iron level is not lowered, and the reason you can understand. I mean, for a plant, it's not advantageous to put less iron in the seed, because then these seeds cannot germinate well. Instead,
yes,
the strategy of the plant becomes, I should produce less seeds, but make sure they have enough iron to successfully germinate.
Well, if there is something wrong, is not happening.
Yeah, I mean, some plants also have this strategy, but some plants not. And we don't know, I mean, what causes this strategy. And of
So
course,
now I think Babar is working on this, huh?
yeah, so we have one mutant which has low iron and then germinates later. And now we want to generalize this, like if seeds have low iron, then they germinate later. But to check it, there are not so many mutants that have low iron. So this one is special for us.
Yeah.
Okay.
Should I pass this?
Yes.
So far, they have observed that so accumulation of iron in those mutated seeds are compromised. So they, in figure 4, they just wanted to check whether common biosynthesis in mutant seedlings are affected or not. So in part A, is these two genes, IRT1 and F402, are involved in iron uptake, and both of their iron deficiency induced expression are reduced. And in part B, we are looking at the genes that are involved in the common biosynthesis, and this is the crucial part. So this gene is the one involved in the, this is the first one involved in the common biosynthetic pathway, and this gene is responsible for fraxitin synthesis, and this one is responsible for staterin synthesis. So again, we see that their iron deficiency induced response is reduced as they have speculated. So in part C, again, we see the genes involved in coumarin secretion. Again, this one's iron deficiency induced response expression was reduced. However, this one doesn't change that much. So in the part D, we see the genes involved in control of coumarin synthesis and secretion. And again, we see that their iron deficiency in this expression is reduced. So from this, we can just say that BHLH121 just positively regulates fraxitin and staterin synthesis and secretion. They just concluded that from this. Is there any questions?
Yes. You know what I'm gonna ask.
Hocam, because they just saw that iron accumulation is less in the mutants, they just wanted to check whether these iron uptake is compromised or not. So they did this. What I said it.
Sorry, I then missed. So why did they look for the expression of genes?
Because their expression levels will be induced when iron is not present due to stress conditions. So plants just want to take up more iron, for example, when it's not present. So we can just compare them with the control conditions. And
Yes.
if it's reduced in the mutants, we say that it's compromised.
It is compromised.
It's reduced or impaired.
Okay. Is Irem using the word compromise in the correct way or not? I'm not sure. Does it mean it is lower when we say it is compromised?
Hocam, it doesn't work normal sense. It doesn't work in a normal sense. It means, as far as I know. Yeah, but I'm not sure. If anyone knows, please tell.
Okay. Yes.
So
Why do they check these coumarin biosynthesis? I mean, they already check and look the expression of IRT1 and FRO2.
hocam, because these coumarins are the compounds that making the iron uptake more efficient. So maybe if those are impaired or compromised, the plant won't be able to take up iron that much as the wild type.
So they're examining the mechanism behind why the mutant is iron deficient.
Yeah.
So they first look fit-related genes to find out the mechanism, and then they saw IRT1 and FRO2 are compromised.
Yes,
Okay, and then these were genomic level, I think. They also want to see in proteomic level, protein level, if something is wrong, and then they look to the these chemicals, and then they see, yeah, these are also problematic.
Yeah, we will see the coumarins in the next part too.
Okay, so
Experiments related to them.
they are trying to explain why the mutant is chlorotic.
Yeah.
And cannot take up enough iron. Okay.
John?
Yes.
In part C, they showed that the PDR9 like expression gene wasn't affected, and they said that it wasn't affected, but they don't really explain why.
Yeah, I'm not sure why either.
I think, and this is a theory I'm not sure, but I was actually just interested in what your opinion is. I mean, these expression genes are the ones that are expressed more when iron is lacking, and it's like a response. So maybe this is not related. That's why. But otherwise, I couldn't find a reason why.
So what you see is PDR9 is not expressed lower, or what did you say? Can you tell again?
There, the expression in response to iron deficiency was not significantly affected in the nutrient compared to the wild type.
But I see it looks like it's affected. So
I mean,
these are
not
affected,
significant.
but this one is affected, right?
Hocam, they said in the article it's not altered that much. So they just said it's just not
they didn't
changed,
really
not
give
affected.
a reason why.
Okay, which ones are affected? Which ones are not affected? Don't
mean,
see what they say. Just look at the data.
SNH, for example, is very much affected, but BGLU42 and PDR9 are not really affected.
I mean, this one is now lower. The others are also lower, but not that much. So for me, it's not so surprising. Since this is lower, that's okay to me. I don't know. So it can be anything. Maybe there are some feedback mechanisms. Maybe this PDR9 is secreting scopolamine, and if the plant doesn't have a lot of scopolamine, maybe this tells the plant do not activate PDR9. Maybe this is the reason. And now I notice, so under iron sufficiency, we have this expression level. And under iron deficiency, we have also almost the same expression level. So this gene is not really overexpressed upon iron deficiency, really. You see, they are also similar. So I wouldn't expect the mutant to have much less. Did you get it?
For this one, do you think that this is real?
I don't know. Why not?
From the previous other people's experiment about this PDR9.
I'm not sure. How was it?
Yeah, me neither, but maybe this is correct, but maybe the experiment itself is wrong.
Yeah, you know, some genes are affected a lot. MIP72, I'm sure it's affected a lot, and MIP10. from iron deficiency. You can see the difference. But PDR9 maybe not one of those genes.
Yeah, Hocam, that's kind of why I was interested in asking, because I read in the article and didn't really explain why, so that's why I was kind of interested.
Good, very good, Abdullah. You go for details, and it's a very good point. I mean, then we can discuss about it and we can get deeper into the issue.
Yeah, thank you, Hocam.
Yeah.
Should I continue?
I think so.
Hocam, in part B, they have just look at the genes that are involved in coumarin synthesis. So in part E, they just want to examine which coumarins are accumulated in the root extracts, and they have used the absorbance chromatograms, which are obtained at 338 nanometers. I think this wavelength is the one coumarins are detected much, so they just use this. So this experiment is also conducted at pH 5.5, and this is the pH at which cystatin biosynthesis and secretion preferentially occurs compared to others. And here we see the ones that are accumulated in the root extracts and their names. Here, this is the biosynthetic pathway of coumarin synthesis, and this is the first gene that is involved in the coumarin conversion. So when we look at this one, this is used as negative control. And since this gene is mutated, we don't see any accumulation of these coumarins. For this part, actually, I searched a little bit extra and I found an article. They say here scopoletin, fraxetin, and cystatin are found as glycosides in the roots. So glycosidic forms are actually inactive forms, and we expect to see glycosidic forms here. And the deglycosylation likely occurs before the export of them to the rhizosphere. So when we look at the wild type, when iron is present, we see the accumulation of scopolamine in high amount. However, when we look at the wild type, when iron is not present, we see both cystatin glycosides, scopolamine, but in addition, we see cystatin. But here, I would like to actually ask you why we see accumulation of cystatin if it's not in glycosidic forms, and why is it accumulated in this active form?
Okay, I think one of the students should answer this question before we tell something.
I don't know the answer.
You can ask the question again.
So I'm just a little bit confused. For example, in wild type, iron is not present. We see these glycosidic forms. However, in this part, we see the cystatin accumulation, but it is the active form as far as I know. So why do we see the accumulation of this cystatin here?
It's the active form.
Maybe they just write it wrong, but they just write cystatin for the fourth one. So I'm not sure.
Why do you think there shouldn't be cystatin?
Because it's active form and we are just looking at the root extracts, and these are not yet secreted to the rhizosphere. So they should be in inactive form. But I don't know that, for example, if deglycosylation occurs in the roots just before the secretion of them to the outside, or deglycosylation happens and the active forms also can accumulate it in the roots. I don't know this.
Yeah, I think nobody knows that.
Okay, so this is
they are trying to understand how this activation occurs, especially for scopoletin and scopolamine. This is an active debate. So it's okay. You find this active form. It is gonna be secreted. Yeah. I don't know. We don't know the answer. I think there is no definite answer for that. I think, as far as I know.
But from this paper, we know something. We know that under iron deficiency, these genes are highly expressed, suggesting that this entire metabolic pathway is supposed to be shifted from the precursor to the final product. Therefore, in iron deficiency, we would expect to see the cystatin accumulation. And actually, glycosylation happens in the Golgi, it starts in the ER and it finalized in Golgi. This is known. And actually, for instance, for sideretin, it will be glycosylated and it will be stored. And there are some enzymes that can remove the glycosides, and then immediately it will be secreted out. But we still don't know those enzymes.
Okay, hocam. Thank you.
But
And also,
I think this is also secreted. This is also
Yes, fluoxetine is also, sideretin is also, scopolamine is also, and actually, most probably they, now, as you can see in their structure, they have OH groups.
Yes.
And this extra oxygen that can interact with something else in the cell, cytosol. Therefore, they are glycosylated from those areas and then they are stored. So this is just showing the active protein, the active, what is that, sugar molecule.
hocam, can I ask something too?
Yes.
Hocam, here, as we said, here sideretin is accumulated, but we don't see any fractionated. So maybe are there any unknown enzyme yet, for example, just synthesizes sideretin from scopolamine?
It is very possible. There might be another CYP protein over there doing that. That is maybe possible. Or maybe somehow the concentration of the fluoxetine is much less compared to the other ones. Therefore, they couldn't detect it.
Okay, hocam.
Hocam,
By the way, yes, please.
Thank you. By the way, they did this experiment for the root extract, not the root exudate. So that's it makes sense that we see the inactive form of the sideretin.
Yeah.
Yes. Good point.
By the way, I sent you a link, a paper about the expression of the PDR9 under iron deficiency.
Yeah,
in the image, they showed that it is highly expressed under iron deficiency. So now we need to question this paper.
Yeah, you're right. By the way, why do they provide a lot of information about these chemicals?
Hocam, they didn't. I found them.
I mean, why did they put these things? I'm also asking everyone. I mean, I think they are not needed because it's not related to the study. The study aims to find the interaction of these two transcription factors. But this is just a normal response of iron deficient plants. It doesn't really provide any insight about the interaction itself, I think.
Now, if you ask me, I think they are trying to tell that this BHLH121 is affecting the coumarin synthesis.
Hocam, yeah, they said this data suggests that BHLH121 is a transcriptional activator of fractionated and sideretin biosynthesis.
So I think they are somehow trying to make a new opening, new door they are opening, so that in the future somebody will work on this type.
Isn't this interaction through FIT?
But actually this BHLH121 doesn't interact with FIT, so most probably it is interacting with something else. There is another pathway they are trying to say later.
Okay, then I missed it, that point.
Right, it was not interacting with FIT.
Yes, hocam.
But isn't the knockout of this, in the knockout of this, is FIT still active? I think FIT is not active anymore.
Hocam,
did they check the expression of FIT?
Yes, hocam, they check BHLH121 binding to the promoter of the FIT, and it doesn't bind.
Yeah, maybe it doesn't bind, but something else binds, which is affected by BHLH121. So if FIT is not expressed in the BHLH121 mutant, then it would explain all the things, and it is not interesting anymore, I think. I mean,
yes, you are right.
FRO2 and
IRT1
IRT1
highly expressed,
and their expression is also abolished, I think. Can we look back to the FIT and FRO2? Sorry, IRT1 and FRO2 expression. Yeah. So you see, they're regulated by fit, and I think this shows clearly fit is not active. And
Yes.
if it's not active, all the things, this metabolic thing will not be, of course, working properly. So I don't see the relevance of giving so much attention on the metabolics.
I'm passing this, hocam.
Okay.
This is just the reference, the article that I found for this information. It belongs to just 2018, and it's a very nice article actually. If anyone just wants to read it, they can just, I can send it to you later.
It's from my professor.
Really?
Yeah, Nicolas von Veren.
Oh, I didn't check this, hocam. It's
Yeah.
nice then.
Yeah, it's very, very, very good. State of art article, also how it is written.
In figure 5, they just aimed to measure the expression levels of genes known to be involved in iron homeostasis in BHLH121 loss of function mutants. So here we can see that all genes iron deficiency induced expression was reduced except BTSL2 and IMA3. Other than that, all of these genes expressions are compromised. So they are not expressed properly, or not at all. Should I pass this? This is the only conclusion that they have made from here.
What's the reason of having this?
Hocam, they just want to know whether BHLH121 loss of functions affects any of these genes expression levels, so they just measured it.
Why do they want this information? I mean, they looked already for IRT1 and FRO2.
Hocam, because all of these genes are known to be involved in iron homeostasis, and the overall aim of this article is to just show BHLH121 transcription factors effect on the iron regulatory network.
Yeah, but they also look for IRT1 and FRO2. Wouldn't it be better if they put those genes here instead of showing them earlier?
I don't know, hocam.
I think they try to show this BHLH121 is directly affect the other genes, but for these ones it's not direct, but they try to show there's another relationship in somehow it affects these genes.
Yeah, I think IRT1 and FRO2 are proteins, but these are all regulatory things, transcription factors. So they want to show those regulatory things themselves are regulated by BHLH121, which means it's upstream of all those known regulatory genes. So it's the master regulator.
For the others, right?
For the other regulators. So you have the in Turkish müdür, executor, and then you have the chief executor. So here the authors compare the executor's hierarchy. So putting IRT1 and FRO2 here would not fit.
Yeah, but by the way, also it's written in the article, they said BHLH121 acts as an activator upstream of FIT. I looked for it, and I think this upstream activating sequencing is something distinct from the promoter, but it's also affect the expression of the gene. And also they said activator upstream of it. I understand that this BHLH121 is binding this activator sequencing, upstream sequencing of fit, but I don't know if it's true or not.
I don't know. So your question is it possible to have a activator sequence that is not inside the promoter?
Uh-huh.
I
It
don't
is
know.
possible.
Mhm.
This type of thing can happen.
Okay.
I'm passing that.
Yes.
Okay, in part B, these are the genes that are all possible targets of the BHLH121 transcription factor. So they just measured the DNA binding ratio of BHLH121 to these promoter regions of those genes. And this experiment is done with the method ChIP-qPCR. And here we see the complemented BHLH121 two lines. These lines, actually this line is used because in this construct, growth defects of the mutant is rescued. So here I just understand, for example, that they chose this BHLH121 over the other two, but I don't know the reason why for this. So since the growth defects are just rescued, they just use this complemented line. So to this construct, they have also added GFP protein because for the ChIP assay, they will use anti-GFP proteins so that the chromatin part of this construct can be taken. And if this BHLH121 transcription factor protein binds to any of these promoter regions, with qPCR, we can amplify them and we just can find if there is binding of the BHLH121 to any of these genes or not. And in the end, they just saw that BHLH121 does not bind to FIT, F6H1, S8H, or CYP82C4. But other than these four, BHLH121 is able to bind all of these promoter regions of those genes.
I have a question.
Yes, hocam.
If this gene 121 cannot bind to the FIT promoter, how do you think that the expression of that is different in the mutants? In the mutant, you showed that the FIT expression is different in the previous image.
Mm-hmm.
Yes, it is very low in the mutant. And then you show that it doesn't bind to the FIT promoter. So it seems that there is no interaction of these. Then how do you think that the expression of the FIT decreased?
Hocam, isn't that just shows whether these BHLH121 and PROFIT interacts?
Okay.
I'm not sure.
It tells that the BHLH121 doesn't bind to the FIT promoter, okay?
Mm-hmm.
But then you have a different expression of the FIT gene in the mutant. What causes this difference? So definitely this doesn't happen because of the BHLH121.
Hocam, can there be a BHLH121 analog in the cell that mimics the BHLH121?
I didn't understand, sorry.
Is it possible that there is a BHLH121 analog in the cell that mimics BHLH121, so activates FIT, maybe? Did you understand?
So something else is interacting with FIT for sure, but it doesn't need to be analog to BHLH121, I think.
I mean, Ajam, they mentioned that BH21 acts upstream and it's unlikely to interact with the promoter of FIT. So it probably is a form of network interaction. So it interacts upstream of FIT and then the network just kind of follows through like a memory. So it affects something that affects something like A signaling cascade is the word I was looking for.
Maybe this BHLH 121 affects the other transcription factors expression, which they all in the fit expression, I think.
Yes. Can you give an analogy to this?
Like it's a little bit hard right now. Actually, there is some transcription factors which affect the expression of fit, which binds the promoter of the fit, and these are the I forgot their names, but I think BHLH type. I think 101 is binding the promoter of the fit, if I didn't remember wrongly. And this 121 affects the expression of these transcription factor, and this transcription factor cannot bind the fit promoter, so that the expression of fit cannot work well.
So maybe the analogy would be just imagine BH. So 121 pushes someone, and then this guy whom he pushes it just hits to the fit, and then the effect happens. I mean, fit falls down. It's not like 121 directly pushes fit, but it pushes someone to the fit.
Hocam, should I pass this on to?
Okay.
And Mojam, did you expect this answer, or you actually ask it about, you don't have any idea about it?
Now, we have seen the phenotype of the mutants, and then that phenotype is supposed to be produced because of a gene expression difference. So bottom line was that even if there is interaction between the two transcription factors or activation of one another, it really doesn't matter, because the entire system is messed up in the mutant. Therefore, that messing up with the amount of the iron in the plant is affecting the expression. So it is not because of the binding or not binding of the BHLH 121, it is just the amount of the iron affecting the expression level.
What do you mean? So in the mutant there is less iron, so fit should be upregulated normally, but there must be a mechanism that prevents this upregulation.
Yes.
And since 121 does not directly bind, then it should make someone else bind to fit.
Yes, there is something different that they didn't get into the detail of it.
Yes.
Hocam, can I ask something?
Yes.
Yes.
Is it possible that this experiment is false? Because I have a presentation 2 weeks later, and in my article is also about BHLH 121. And in my article, it says BHLH 121 is directly binds to fit promoter. And they did 4 experiments about this. It's here. And I'm really confused.
I wasn't expecting that.
You see?
Okay, so this shows us, Eylül, now it's for you very important to listen very carefully these slides and these discussions, so you can show us all the things that are not common or common in both papers, and what is contradictory, you know. So you tell us that's there is something contradictory. Yeah. about fit binding.
Okay.
It's very interesting point.
Hocam, I'm passing the figure 6.
Yeah.
In figure 6, their aim was to check whether BHLH121 gene expression and protein localization pattern is correlated or not. Bir şey mi dediniz hocam?
Yok yok. Dedim.
Through figures A to C, we will see the expression pattern of BHLH121 gene. So in part A, we see that the gene is expressed in all of the given plant parts. So we can say that BHLH121 expression is not restricted to any specific plant organ. In part B, they measured the BHLH121 expression in different iron concentrations. And as we can see, they didn't observe any significant alteration in expression of the gene. So again, they concluded that the expression of BHLH121 is not affected by any changes in the iron availability. In part C, they just visualized the BHLH121 promoter activity. Actually, how they did this is firstly they just cloned the promoter of BHLH121 gene, and then they just fused it to Gas reporter gene, and they produced this construct. So this means the Gas gene is under the control of promoter of BHLH121 gene. So whenever BHLH121 is expressed, we will also see the Gas reporter gene will also be expressed, so that we are able to visualize all of them in different plant organs. And Gas activity is also confirmed that omnipresent expression of the gene. So it's present in all types. Any questions here? I'm passing it. In part D, these are just the sections from the root differentiation zone, and they just tried to visualize the protein localization of BHLH121 this time. So it's done by producing a construct of BHLH121 and green fluorescence protein. So in control conditions, we see that GFP fluorescence is detected in the nuclei of the cells located in the central cylinder here and endodermis. However, in iron deficient conditions, we see that this fluorescent signal is detected in rhizodermis and the cortex. So just to further confirm what they have seen here, they just did the part E. So they just compared.
Sorry, may I ask something? So we said this transcription factor should be in the nucleus always, and now you tell us the position of this transcription factor is changed.
Hocam, it's not changed. It's in the nuclei. I said just the plural form.
Okay. I just wanted to confuse you. So yeah, the cells are changed, but still it's in the nucleus of the cells, right?
Yeah.
Okay.
May I ask a question? Now, when there is enough iron, so you find this protein in the middle part of the root. And when there is no iron in the environment, suddenly you start to see more of this protein close to the outer part of the root. Do you think that a transcription factor can move from one cell to the other cell?
Hocam, transcription factor cannot move, as far as I know, but also the researchers doesn't know why this fashion happens. And I will just show a slide for this. They have two hypotheses for this, but they don't know which one is correct or any of it's correct or not.
So this Emre Hoca's question was a very relevant question, right? They try to answer the same question.
Yes, hocam. Should I pass?
Yes.
Okay. In part E, they just wanted to confirm what they have observed in part D. So for this, they have compared the percentage of cells which are GFP positive and to the total number of cells in these cell layers. So here, the orange ones represent the unlimited control conditions of iron, whereas the green ones represent the limited iron conditions. So as we can see, when iron is limited, we see the BHLH121 protein is located in the rhizodermis cell layer and cortex cell layer. However, when iron is not limited anymore, we see that BHLH121 protein is located in the stele. So it's a very interesting finding actually. So data of DNE part suggests that the location of BHLH121 within the roots depends on iron availability. So as I said, they don't know why iron availability modifies the localization of BHLH121 to these specific cell parts. So in article, they have just proposed two hypotheses for this, but they didn't check and they don't know whether one of them or any of them are true. So firstly, they thought maybe some post-transcriptional mechanism could modulate the stability or translation of BHLH121 mRNA in cell type and iron-dependent manner. And secondly, they thought maybe some post-translational mechanism regulates the BHLH121 localization or cell-to-cell movement. So both of them really make sense, but as I said, they don't know whether or not they are correct or not.
Can you tell with your sentences what first and second hypothesis tells?
So after BHLH121 is produced, it's exposed to it's exposed to post-translational mechanism in part 2. So this post-translational mechanism's results maybe regulates the localization of this protein. So maybe it just changes, for example, let's say BHLH121 protein is, let's say, phosphorylated maybe, and it's able to enter some cell types and not able to enter the other ones. So post-translational mechanism just changes.
Can you tell the difference of two hypotheses in one sentence?
They are just different in post-transcriptional and post-translational mechanism.
Post-transcriptional, post-translational. Okay, maybe I misunderstood. So,
Which part, hocam?
So according to what I understand, first one tells the protein in the cylinder is destabilized and synthesized again from scratch in the outer cell layers. The second hypothesis says there is no destabilization or new creation of protein, but it is transported to the outer parts. Do I understand correctly?
Hocam, I'm not sure about the destabilization part. I don't know.
Trying to say, in the first one, new protein is generated in the outer cells. In the second one, the same protein travels to the outer cells.
Yes.
Yeah.
Okay. So if you cannot make it short, you don't understand it. I think this is a general.
Yeah, I didn't understand that part very well, so I just directly write down their hypothesis for both of them.
Okay.
Sorry for that.
Yeah.
In figure 7, we have finally a proposed model, and as we can see, BHLH121 just acts upstream of the iron homeostasis regulatory network. So BHLH gene, along with the ILR3 and its closest homolog genes, produces some proteins, and these proteins are able to form heterodimers, as we saw before. And this heterodimer is able to just directly or indirectly activate the genes involved in iron homeostasis network, and this is the regulation part so far proposed.
I like this drawing.
What?
I like this last figure.
Mm-hmm.
So I just wanted to give you a summary of observations, but if you want to, I'm not going to go over them again. So this is just the end. Thank you for your attention. I felt like this while I'm preparing this presentation. So thank you all. That was all.
Yeah, thank you very much for the presentation.
Yeah, it was a nice presentation. By the way, I want to ask a question to everybody before we finish. Now, imagine that there is a protein in the vascular tissue in the root. And then after you do a treatment, then suddenly it shows in the epidermis. Do you think that it is easy or very easy for a protein to move from one cell to the other cell in the root?
It shouldn't be.
Why not?
Because they have to be,
No, sorry. Go ahead.
they just have to be regulated strictly. If any of those proteins are easily accessible to any of these cell parts, the mechanism of other pathways would be disturbed.
Usually also proteins have tags on them, so they have to go to a specific location to be part of a specific segue, like a part of specific pathway. So they would have to change the tag on it to make it go to a specific location.
Now, for instance, if you are thinking of a movement, let's say of iron that is uptaken from the rhizosphere into the epidermis, and then it needs to migrate from epidermis to cortex, from cortex to the endodermis, pericycle, and finally to the vascular tissues. But there is one problem in this movement. There is a huge barrier in the middle. What is that? Do you know the name? Yes, Casperin strip. Now, do you think that a protein, let's say a transcription factor, can migrate from one cell to the other cell through this Casparian strip?
Yeah,
Well,
but
it can,
yeah,
but how?
simplistic pathway.
So it will be very difficult, I think, if you ask to me. And it should be extremely regulated.
So isn't the system like a vesicle takes it, the protein? And since this is, yeah, okay, in the vesicle we have the protein, and then this vesicle can travel through simplast maybe. I'm not sure if vesicles travel through simplast.
Hocam, what about it is not being transported but expressed by another transcription factor or another gene? So in one condition, it is being expressed in one part of root, and at zero iron, it is being expressed in other part of the cells. I guess this is the point, because this is a protein.
I think they have two hypotheses. One of the hypotheses is what you say. So the cells express it de novo. So, but
Most probably it is being expressed.
yes, but in the other hypothesis, they say the protein travels.
They say there's a post-translational modification.
Yes. So proteins can travel inside within a cell with vesicles by just diffusion, or somehow with this cytoskeleton, maybe directly. I'm not sure. But
Now,
I don't know
I think
how they.
Babar might be really true. Actually, in this experiment, they were using the native BHLH121 promoter gene construct.
Yeah.
So actually, it is not only showing the protein accumulation, it is also showing the expression of it. So under iron deficiency, it is more expressed and accumulated in the epidermis, for instance, which may suggest that it is itself might be regulated by fit.
Yes. This is one hypothesis.
Instead of moving from one cell to the other cell, which is actually difficult, maybe expression is just changed and it is just that easy.
Hocam, if we want to test out these two hypotheses, which one would be better? I mean, what experiment would we design to test out which hypothesis of these two would be better? What would be the experiment that we would design?
I think you should have the microscope continuously recording, and I don't know if it is technically possible, then if you see continuous movement of the vesicles or proteins.
Brownian movement?
What?
Brownian motion, maybe?
It doesn't have to be Brownian motion. I mean, maybe it's in a vesicle and then it travels from cell to cell. It just moves like this under the microscope. You may be able to see.
Could we tag the protein in any way?
It's already tagged with GFP.
Yes.
That's why you can see it under the microscope. Actually, in confocal microscope, you can observe the movement if it is really moving. You can record movement for several hours.
Mm-hmm.
Or maybe they can look at the expression of this gene in fit mutants, do something with the fit mutant.
Or one other possibility, maybe they can put a chemical which stops de novo synthesis. So all the synthesis stops. And then if we still find change of the protein, then we say it traveled.
Yeah, I think that will be a nice experiment.
And another possibility, if this is traveled and if this is traveled by vesicles, then maybe you can put an inhibitor of cytoskeleton. Then this should also stop it there.
Yeah, I think I have seen this type of papers. Yeah.
Yeah.
Anyways, I think in future they will work on this, right?
Okay, so if we are finished, Irem, can you stay so I can give you feedback?
Thank you very much.
Yeah,
See you next week.
Like, by the way, you can also stay to give feedback if you want to share some information.
That would be great.
I can wait just for 5 minutes.
Okay. So bye-bye, everyone. Thanks for joining and for your comments. I think we had a lively discussion. I liked it. Merhaba. Ben direkt başlayayım mı?
Olur hocam.
Evet.
Tamam. But I do it in English so I can put it on the website in YouTube. Okay. Can you show the presentation, İrem?
Tamam hocam.
First, yeah. Next one. No, I mean the second slide.
This one.
Yes.
Hocam, I just wanted to give a very basic introduction since Anna is here too. I wouldn't do that normally.
No,
but...
you should give an background. It's okay to give this background, but you should also give some background about ILR and other transcription factors. So you have this picture at the end, you remember?
Yeah, that
This
network.
Network, yeah.
That network,
So
yeah.
maybe just imagine this BH121 is not found, and then you still have a network. Maybe you should put a network without BH121 and then explain some of the important things there, especially the things that come up in the presentation. So we understand what the scenery was before they started. And then we see, now this is changed.
Okay, hocam.
So
You're
this
right. I should. Should I pass this?
Yes, and this third one. So I like the idea you give information, background information, but if you give it like this, it takes too much time, and then people don't know with what to integrate this information. So I would suggest give them the first data, so the fourth slide, okay? And then you say why they do the experiment first. And then when you need to explain the experiment, then you move to the previous slide. Then it will make more sense because now they don't know what they are gonna see, you know. They cannot integrate. In fact, you did it already very nicely in figure 6.
In figure
No,
6, sorry.
figure 6,
yeah.
yes.
Um,
Figure 6C. So in figure 6C, you showed us the Gauss line, the logic of, I mean, the methodology, but you didn't make it a whole picture. I mean, you first mentioned the experiment, and then you told us how the methodology was, and then we understand it perfectly. So you should also use the same technique for the previous ones. Yes, very big problem. By the way, I didn't have time to sıralamak, make it an order, like the most important point to the least important. But this one was the most important. I think the logic was difficult to follow, and there were 2 reasons for it. In some of the graphs, you didn't tell us why they do the experiment. You directly started what they do. And I warned you, like, why they did this experiment. I asked you several times, you know, but then you start to tell about it in the final figures, I think. So you adjusted yourself, but from the start, you should always put emphasis on why the experiment is done.
Hocam, I tried to do for all of this. Actually, I didn't adjust anything. These are my notes from the beginning. I said aims in all times. I'm confused a little bit.
Yeah, you know, Ayşe also was saying I am doing this, but she was not really doing it properly. So if you try to do it, it didn't work because we didn't understand. I mean, we need to build the logic. To build the logic, we need to understand what is before, and now what they taught, why they do the next step. So you should tell us why they do the next step, but not any other step. What was interesting for them? What was the next question? You should always think like, what is the next question and why? So maybe you should work on it. It's not easy to understand the difference, I think, between telling the experiment and telling the logic. But there's a difference. And the second problem for building the logic was the titles were missing. Every slide must have a title. I think I told this, but maybe you forgot, right?
Yes, hocam.
Yes, this was a big mistake for me because I told it, and in the other feedback, if you watch the YouTube videos.
Hocam, I watched your YouTube video, but I thought, for example, just giving one title for this and continuing the figure is enough, but it isn't, I guess.
Okay,
for example, this is the figure 1 and all of them belongs to this, and this is the figure 2, all of them belongs to that one, but I guess it's not correct.
Yeah, the reason is the reason of having the title is when someone sleeps, when he wakes up, he should still.
Okay, hocam.
Yeah, you get it.
I get it. Okay.
Now, Irem, while of course in the paper they have a less place there. So in that limited area, they want to give one general title and then they put several small pieces together in that entire image, and you are just chopping it out. And actually, when you do that, you are separating different experiments, and each experiment should have an aim and a final conclusion. Therefore, it is better to write the final conclusion of that individual experiment in the title. It will be easier to follow for yourself while you are presenting as well as for the audience.
Tamam hocam.
One small problem, just this slide and the next slide, 7 and 8, yes. So here you say, you tell the 7th slide, and then you say to confirm it, they take these measurements, you say, on the 8th slide.
Oh, I said it wrong then. I just wanted to mean that for this one just shows the previous one.
So the
But it-
correct word should be quantify. To quantify it, yes.
Okay.
Confirming is different because you need to do another experiment to get the same result.
Okay hocam.
Yes. And very important point, you have to shorten the sentences. If you are not able to shorten the sentences, this should tell you, this should alarm you, you didn't understand the topic.
Okay.
You didn't understand the meaning of the sentence. Like
In hypothesis
I mean,
part.
the sentence is too long.
Mhm.
Also, these hypotheses of yours,
Yeah.
they were too long. You need to practice paraphrasing skills, I think. In plant physiology, you have this link where there's a video about paraphrasing. I really would like you to listen to the video and develop some paraphrasing skills, so you can find out if you really understand or not. That's all from my side. I should say the presentation is good, and good because I know you had a lot of lectures. And are you still there? Yes.
Evet hocam, buradayım.
You had a lot of lectures, and you didn't have almost any molecular background. You even didn't know like what is promoter, what is gene. I mean, you didn't have a good sense of that, sense of GFP. And when I look at you right now, I think you made a very big step. I think next step now you understand what is going on. But next step is like building a good logic and reflect it to the audience.
Thank you hocam. I'll try.
Yes.
Hocam, Emre hocam, do you have any other feedback?
Nothing negative. I think your last that summary slide, I really like that. It is really summarizing each individual figure, and I would, for instance, put a final summary at the bottom of this slide.
I didn't thought about this. I didn't think.
And while actually you are finishing, concluding, and taking the questions in a real presentation, you should keep this slide open so that people can remember and ask more detailed questions to you.
Okay hocam.
But this was a very good slide, a good idea.
Thank you.
Okay.
Okay. So thanks again, İrem.
Thank you so much hocam.
You did a good job. Please keep up the good work.
I'll try hocam. Thank you. Then see you next week.
Bye bye.
Bye.